Good morning, everybody. Uh, Before even I begin, I would like to start with this. I'm not a native speaker. Oh, sorry. So I'm not a native speaker, so if you don't understand any word that I say, so please raise your hand and then I will repeat that. So I have been studying in the Farm Hub, involving my undergraduate students for last 3 years, collaborating with Farm Hub through Conrad. I began that. And broadly, the study that I did is, the impact of land use on water quality and quantity. So I had to divide my presentation into 2 segments. This is the first one, which I am very passionate about. The other one I also like. But I have a strong feeling that I may not be able to fit everything within the time that I have. But if that is the case, then I may run through, one presentation a little bit, faster than the other. And then I would like to save some time for question and answer as well. So this is, the first study that we did is using a rain simulator to figure out what is the optimum amount of water that we can use to irrigate for our crops. And the other thing that we also try to do, so this will be the sequence of my presentation, just a few seconds. And then the other thing that I wanted to do is we all understand, is not a rocket science, that tilled versus non-tilled land, which one will erode more when we irrigate? Of course, tilled land will erode more. But I did, a lot of, Google search and things, and I could not find what is the real magnitude of benefit of that. So I thought, using this, I might be able to, find a way to quantify that. And that was one of the things that we also tried to do that. So, and the other thing that, the Farm Hub provided for us is, because in order to do an experiment like this, we have to find a place where, the soil type is constant, and then we have tilled versus non-tilled, as well as the crop type is constant as well. In other words, they remain the same. And Hudson Valley Farm Hub, in fact, provided that unique opportunity for us to do it. And that's why I jumped into it. So what we did, we used a soil infiltrometer, developed by Cornell University to, simulate rainfall at a certain rate. And then, this is, a ring approximately 1 feet diameter, and then we inserted that 6 inches into the soil, and then we simulated rain on top of that, placing this here. And then we monitored, what is the rate of infiltration over time. And at the same time, whatever does not get infiltrated, that will run off from here, and then we collect that into a bucket. And that is the runoff water that I used for my second, purpose that I had for this research, or the objective that I had for the research. So what we did, as soon as we started, the experiment, rain started to fall on this ring. And then every 3 minutes, we recorded, how much went in, and then the runoff volume when it started, and we collected that as well. And then when, the runoff volume reached, a steady state or equilibrium, then we stopped the experiment. And also we collected soil samples, to analyze for grains, grain size analysis, just to see, if they are also similar in different plots. And what we did, the total collected runoff that we had at the end of the run, we analyzed those for how cloudy the water is, using a turbidimeter. And the more cloudy implies that more erosion happened in the soil. And so this is the data sheet that we had. For example, we started, what was the height? And this height, in fact, dictates, the head. That means what will be the rate of precipitation through those, infiltrometers. And then when it reached an equilibrium, then we stopped the experiment. Usually, at least half an hour or more for each experiment. And we also, this is, the cloudiness of the water that we measured, using turbidimeter is, and I will mention about the dimensions. So we did that as well for every run that we had. Sometimes we, used 4 infiltrometer at one time on a plot. So this is, I will show you a couple graphs. This is for a non-tilled corn plot and a tilled corn plot, and then this is the rainfall rate, and this is the runoff, and runoff minus rainfall will be my infiltration. And at one point, this infiltration capacity will reach an equilibrium. Even if you add more water, it will not get into the soil. It will either run off or it will evaporate. So for example, now, if we irrigate more, assuming that it will be helpful, it will not be helpful. That water will not reach the root zone. So this is, then we can in fact exactly figure out what will be the optimum rate of irrigation for this particular soil type. And this is the advantage of this experiment. So for example, here, we are not worried about water because due to global climate change, we are getting more precipitation, but think about, west, southwest. They would be benefited with this experiment a lot. So the other one that I would like to show you for a tilled bean plot and non-tilled versus tilled. So each will have, there will be difference between these because soil, flow through soil in unsaturated medium is very complex. So for that, we need to do more experiments to come up with one number. Eventually, at the end of the day, we will say, okay, this should be the rate of irrigation. Let's say hypothetically, 1 centimeter per 2 hours, for example. So this is, the result of the cloudiness of water after each run. So this is corn, tilled versus non-tilled, beans, broccoli, and these are the ranges of turbidity that we encountered, tilled versus non-tilled. And through this experiment that we had, we ran probably, I think, more than 40 or so that are meaningful. And we found out that after taking all the averages of this, that tilled fields had water 17 or approximately 18 times more, soil particles than the non-tilled. So at least for this particular soil type here that we, studied, we can safely say that non-tilled land, in fact, 18 times better in terms of soil erosion. But the limitation of this experiment is this cannot be transferred to another plot using this. Then we have to come up with another experiment to figure out, okay, how beneficial that will be. But at least for this scenario, this is much better. But at the same time, sometimes, non-tilled plot may not yield, a good crop compared to tilled. So there has to be a balance about that as well. So for example, now, if we know what is the optimum rate, and if we irrigate this way, we might lose water. Although, we have plenty because for farm hub, we can, pump from the esopus, and there are good amount of water. But wherever, the water is not that, available or scarce, this would be a good way to figuring out, okay, what is the optimum rate? So this is an example of, tilled versus non-tilled, but we did not run experiments like this. We tried to dip, when this became stable, especially the tilled, and then we started running experiment on those. So in summary, so erosion of tilled land is approximately 17 or 18 times higher than non-tilled. So the equilibrium infiltration capacity that we found out, these are the numbers, 65 centimeter cube per minute and 55 centimeter cube per minute for tilled versus non-tilled. So there is not significant difference there. And the average equilibrium infiltration capacity for all the runs that we had is approximately 60 centimeter cube per minute, which is equivalent to 1.32 millimeters per minute of rainfall. So, and if we do more than this, after a while, then we will be losing that water. And that is the point I'm trying to make. Like, okay, then why? So then why should we irrigate more? I think that is my last slide. So what I would like to do next summer is I would like to use a lysimeters, which can collect water samples from the root zone, in unsaturated zone. So this is a porous cup you can insert and then create a suction, and then monitor the nutrient front through the soil. And that's why I asked the question to the microbiology group, is what role these microbes play in the nitrogen cycle. So that will give us a little bit more information about that. At the same time, by running this experiment, we can in fact come up with a term that is called TMDL, total daily maximum load for fertilizers, that should be good for this type of scenario. And this is one, so one thing I wrote down here is overcoming instrumental limitations. The soil infiltrometer that we used is so finicky. For example, if we do it shaded versus sunny, the rate differs. If we use cold water in the morning from the faucet versus room temperature water, the rainfall varies, because what it does, it creates air bubble depending on the temperature, and that changes the pressure of the unit, and which controls the rainfall rate. So we have to do this a little bit more to figure out what will be an optimal condition for our future runs as well. So I'd like to thank you many, Anne, Conrad, Jeff, for helping me with this, and my students. In fact, my son also helped sometimes. And then Bob, who is a funny guy from Cornell, he's the director of the Soil Health Lab, who also trained me how to use this and gave me some clues, how to run this instrument properly. So that is my first one. And how am I on the time?
You have 6 minutes.
6 only? Okay. Can I go for 10?
Yeah, I think we're fine.
We are fine?
Yeah, don't worry.
So the second, presentation that I would like to share with you is the other thing that we did. We collected samples from different locations from the Esopus Creek, Lower Esopus, and some tributaries, and then we monitored, what is the change of concentration over time. So, and the purpose of this, to figure out, this new farming technology, or especially the environmental friendly fertilizers that are being applied in the Farm Hub, how effective that is in terms of water quality. So I would like to begin, this one is, one of the things that Anne mentioned in the email, that, you should highlight, the relevance of your presentation with other, presenters. So the quality of water and the quantity of water is related to all the other experiments that are being done for this Farm Hub. So this is relevant in all aspects. So it is obvious that we need more food, and in order to do that, especially plant-based food, we need to irrigate more. That is the bottom line. And also we are applying agricultural chemicals that will also affect the water quality. Again, this is the sequence of my presentation. I may have to run very quickly for this one. because everybody is waiting for the food, I think. I'm just joking. So I don't want to talk about, Hudson Valley Farm Hub. We know what the role they are playing, an excellent role. So I'll just quickly go through that. So the hypothesis that we are testing here is the environmental friendly farming practices should improve the water quality in and around Farm Hub. So that is the hypothesis we tested. And then what we did is the changes in water quality is compared with 2008, a study that I did with my students, and there are few locations, 4 locations that coincided with the work that I've been doing now. And then 2017, 18, and 19, what is the changes of water quality, especially in the context of nutrients, nitrate, and the other parameter that I will be sharing with you is the total dissolved solid. Although I measured a lot of things, and there are different aspects of the study that students did that I'm not sharing here. So what is the importance? I think many of us are familiar with the concept of eutrophication. More nutrients in water, more growth of algae and plant-based material in the water, and when they die, they use up all the oxygen that we have in the water because bacteria, feeds on them. And if that happens, then we have an anoxic environment, no oxygen in water, that is very hostile or even unlivable for other organisms that live in water. So these are one picture from one of my study area. So look at this overgrowth here. That is, I think, the sample location number 14, and this is 15. So although we are lucky because this is a flowing water, so it will not be there for a longer duration, but had that been there for a long time, many things will die here eventually, or they will leave that place and find a place where they can live. So what we did, the sample location numbers are a little bit weird, but that's how we did it. When I first did this survey with Conrad. So we picked locations, one extreme urban and then one relatively non-impacted. So this is my non-impacted, which is close to the Lamanville gauging station, and the one here, which is in Kingston. And then in between, and there are few, those are small tributaries that contribute also and flows through the Farm Hub. And then the main purpose is to improve the ecosystem of in and around Farm Hub, especially. So these are some of the instruments that I use. Flow meter I used, and I will explain that. This is dissolved oxygen, pH conductivity, this turbidity meter that I also mentioned in my soil spectrophotometer, and the ion chromatograph for common anions and cations. And the flow meter I used is one thing that I found out. If we compare water quality parameters from year to year or even from month to month, if you assume that the same amount of contaminant that is there, and if water increases, then the concentration should decrease. If water decreases the volume, then concentration should increase. Now, if I want to compare from year to year, then I need to also come up with a way so that I can compensate this for volume of water that is present at that moment. So that was a challenge. So we did, in fact, measured flow also when we did these experiments, at least for 4 or 5 selected locations. So these are my students who are working inside and outside, Olivia and Alexis. And also we measured the stream flow in different segments, and this is the way it is done. We have to divide the stream into small segments and measure the velocity using a flow meter, and then velocity times the cross-sectional area gives me the volume of flow for each segment, and if we add those, then I get the total flow on the stream. So this is the way we collected that information in the field. So now I'm sharing only few selected data. So for example, as you can see here, this urban area stood out, and this total dissolved solid means all the chemicals that are present in the water. And then the second one is this 12, which is also an area where there is a development, up gradient, but rest are very similar, not that difference in this case. And many of these are within the farm hub. So this is nitrate concentration in June that I'm sharing with you is for 2017, 18, and 19. Again, 12 stood out because of that development. Probably for that particular event, they probably released some wastewater from their system into the creek or a small tributary, which flows through like a fall. And then the October, again, these are the two outliers. And then there are few outliers here also. This sample location 5 and 9 and 10 are also in the tributaries, the smaller one, not the Esopus Creek. So they stood out a little bit. And we can talk about this during the question and answer session. And then, so this is the stream flow data. The volume of flow increases this way, total dissolved solid increases this way. There is a significant correlation when we did this just for the month of June, we collected number of data point for site number 1, for example, here. And for the nitrate, that correlation is not that much. However, when we did this for June through October, we do not find any correlation for total dissolved solid or for nitrate as well. Because what I found out that during a low flow condition, if the variation is small, the concentration does not vary that much. So that makes comparison of data very difficult, because my goal was, if I can come up with a normalization constant, then I can compare from year to year or month to month. For example, let's say if the flow increases by 10%, concentration decreases by 5%, for example, hypothetically. My goal is to come up with a constant like that, so that I can do this comparison from year to year or from month to month. Again, for site 8, if I use all the data points, then it goes, nitrate shows a little bit, but 0.3 correlation coefficient is not good. But this is the interesting one, so I saved this for last. So in 2008, the study that I did, these 4 sample locations I did in 2008. So sample number 8 is the Lamontville gauging station, which is relatively non-impacted. Sample number 18 is in Kingston, which is an urban signature. But sample number 13 and 16 are within the farm hub. And if we look at the nitrate concentration from 2008 versus 17, and 18, and 19, as we can see here, especially 13, there is a significant decline. 16, after 2008, the decline was also a little bit obvious. But these do not show any relationship. So my takeaway for this is, because 2008, the traditional farming practices was practiced in those areas. But after, Farm Hub took this and they applied, they started applying these environmental friendly fertilizers, my hypothesis still, I'm not 100% sure because I need to do this a little bit more, but the water quality is improving over time in the context of nitrate in this scenario. So, in conclusion, water quality trend shows gradual improvement over time. Even these last 3 years, the trend is relatively good. Either very close to similar or a little bit lower. So variation in stream discharge affects concentration, but I don't know a good way to compare yet. So I need to collect a little bit more, especially now I need to collect some during the high flow scenario. Unfortunately, this year, the flow did not vary that much. Last year it did. So that is, and scientists always say, we need more data. In fact, we do. So we will collect additional discharge data as well as water quality, for next year. And especially, my goal is to come up with, I would like to call this like an empirical constant for comparison, especially for this particular watershed. So I like to thank, few of my students. Of course, Hudson Valley Farm Hub, and Conrad Ann, and especially Sylvia also. She was very helpful. And then I would like to thank, the ACS Curve program of SUNY New Paltz. So what they do, they fund students who are from underrepresented group so that they can do undergraduate research. So I had 8 students from that group, worked with me. And the total, I had 13 students working with me so far. And I will be taking a few more next semester. So the biggest reward that I get for this is my students get real life experience doing things, which is also very meaningful because it is helping the community. So these are some of the references that I cited for this. And thank you.
Questions for Shafiul?
How did I do language-wise? My kids speak on me still. For example, I'm a geologist, so, like earth science, I cannot say art the way you do. It always sounds like art.
I just kind of wanted to ask, how do you think, like with the water studies, how do you think that's going to tie in with the microbial diversity and that kind of relationship? It kind of makes me want to feel like you and my professor should talk and we should collab.
Good question, So, for example, I was going to ask you, did you measure the moisture content when you sampled?
Yes, we did. We have the weather site from, they have a weather station there. Yeah, so we did track that.
I was talking about, the soil moisture content.
Oh, no, but we would like to.
That is important. So I did not want to put you on a spot, but I think next time, when you will be collecting samples, what I would do, I would also, measure the soil moisture content at the same time. And then that will also help you to, find a relationship about, the colony and the number of species, even if you can go that far. But for my next project that I am thinking of doing, microbiology will be very helpful for me, because then I will be able to tie that with the nitrogen cycle and the nutrient front movement through the soil, especially through the root zone.
I had the same question for you. Did you measure the moisture content before you did the rain experiment for the till and non?
So that is related to this what she mentioned, that we had the weather station, so we know the prior rainfall event. So I when we collect water sample, because we collect from the standing or running water bodies. So the moisture content is not important. In this case, whether we had precipitation prior or after might be important.
Well, I was just wondering because it might make a difference till or no till versus how much the soil holds.
Sorry.
Okay.
I thought we were talking about my second presentation.
Oh no, the first one, yeah.
So we did not measure the soil moisture content, because what happens is, for example, if the soil is very dry versus wet, then initially, when we run the experiment, of course, it will absorb more water. So that, the Cornell Cooperative Extension, they came up with a term called sorptivity, but I would like to call that is the initial infiltration capacity. In this case, initial infiltration capacity will be much higher. But when you plot your infiltration capacity over time, it will be like an asymptotic relationship over time. So since my goal is to come up with the equilibrium infiltration capacity, then I don't need to do that. But there is a but. So, but depending on the history of the moisture content, for example, let's say hypothetically, I have rain today, and then I measured my, I did my experiment tomorrow. So the rain is now getting into the soil. So we call it now, it is a wetting front. That means the soil is wet. And when that rain gets into the soil, hypothetically, let's say 20% saturation we have in the soil. Versus another day, when I'm going, there was no rain. It had rained quite a while ago, but now it is 20% soil moisture. We call it in a drying condition. In that scenario, the water front that will move through the soil will behave differently. We call it hysteresis. That means same thing, but behaves differently. So that I did not take into account, unfortunately, and that is very difficult mathematically to solve either.
In your study, did you consider the timing of land application of nutrients on the farmland?
Not yet. Not yet. Next year.
Oh, great.
Next year.
Okay, thanks, Chapter.
Thank you.